Okay, well, let's go ahead and get going here. And this is now chapter 6 here from your book, kind of part 2 of genetics. Get to apply much more of this, of course, now to disease and make sure we have full understanding in that area. First off, let's define two different things here, genetic disorders and congenital defects. Okay, so congenital disorders is the broader umbrella term. That's the big term, and that includes any structural, functional, or metabolic abnormalities that present at birth. So if a baby is born with any sort of abnormality, the key word is here that it's basically there from birth. It's not something that you can acquire, right? It comes from birth. However, the causes are either genetic causes, like through defective genes or chromosomal abnormalities, or environmental factors. So a congenital defect can actually be an environmental factor. It doesn't mean they necessarily have to have a genetic disease, but genetic diseases are a subtype of congenital defect, if that makes sense. Okay. Now, genetic disorders in broad, they can present at birth or they can appear later in life. Like we just talked about Huntington's disease and how sometimes it happens later on in life. Genetic disorders don't always have to occur right at birth. Okay. So let's talk about autosomal dominant inheritance, and let's look at the pattern and then maybe some examples of some disorders. We'll use Huntington's disease as maybe our key option here. So in a pedigree, as you're seeing on the screen here, squares always represent males, circles always represent females. Whenever there's this line in between them, that is I'm trying to think of the I was going to say mating, but whatever you want to call it. It's whenever children are produced here. And that's what we'd call with mice. I was like, oh, I guess that still applies. Here is, and each of these lines that come from that are children from that couple, right? And of course, in this particular case, there's 2 circles, so 2 girls, and 2 squares, 2 boys. Okay. Now, you probably quickly noticed, of course, there's the coloring in on some of these, and that usually indicates they have a particular trait, or they carry a particular allele. In this case, because it's dominant, it would be the exact same thing, right? They have the genetic, they have the genotype, and because it's dominant, they're going to end up having the phenotype as well. So, what you should see very clearly about the inheritance pattern is this. First off, we said this was autosomal, right? What does that tell me? Not on the sex chromosome, so it has nothing to do with it. That means boys and girls, it's going to be males and females, it's going to be the exact same distribution. Now, we look at X-linked, it's going to affect a lot more males, a lot of more females are going to be carriers, and all this difference depending on the male or female. Not the case here. And then we have the dominant, meaning, of course, they're all going to, if you inherit the allele, they have the disease. And then what you see is, in the first generation here, there was one parent that was having the disease, and of course they did exactly mathematically what would happen, and that is 50% of the offspring have the allele. Remember how we did that math earlier? Now, of course, that's just how it works out on average. In reality, it could be all of their children could get it, right? Because it's flipping a coin each time a baby's born, right? So you can flip a coin in 4 heads in a row or 4 tails in a row, absolutely. You could also be none of the kids, right? None of the kids could end up having it. But on average, about 50% of the progeny will be affected in dominant. So keep that in mind, 50% affected. And those who are affected, if they have children of their own, again, it happens again. 50% right down here. 2 of these 4 children end up having the disease. Any questions on autosomal dominant inheritance? Good example is Huntington's disease. Try to think of another good one for you that might be. There's not as many of these that are really well studied. Trying to think, your book has a couple of examples. I don't have it in front of me. Yes?
So with autosomal dominant inheritance, will it always be just like this, like with only one parent's affected, then it'll be 50% of the offspring. So it'll always be that 50-50 ratio if only one parent's
That's right. That's
affected,
right.
versus if both parents are affected, it's 100%.
Well, actually, it wouldn't quite be. It would be, you'd have a 50% chance from one parent. It'd be 75. There you go. It'd be 50% from one parent and 50% from another parent. So it's a pretty bad deal. Only 25% would be unaffected, and 75% would be affected. Yeah. And on the big numbers, it always comes out this way. When we had mice in the colony, I would keep track of all of the breedings. We had recessive and dominant alleles. And sure enough, once you got up to about 100 offspring, it always about averaged out, about 25, 50, 25. Now, when you had just small numbers, like in normal human families, it can go way off, right? All of them can have it, all of them not have it. So keep that in mind. Okay, so autosomal dominant. There's another good one. This is one from your book here, the Brugda syndrome. So unexplained sudden death in a particular individual. What's going on there? They actually have mutations in cardiac muscle sodium channels. This ends up causing a spontaneous cardiac arrest. So in other words, the electrical system of the heart normally allows for a depolarization wave to spread throughout all the cardiac muscle. That system is basically fundamentally set up to all of a sudden break at some point. And here's the reason why. They have a genetic mutation in the sodium channel. Autosomal dominant. His mother and both grandmothers are alive and healthy. What's the son's chance of having the disease? So assuming this is an autosomal dominant, actually, is this an autosomal dominant? This may be an X-linked. Oh, it's also a low penetrance disease. Yeah. Oh, it's right here. I'm sorry. It's very clear. It's right on the screen. Please, someone help read the slide to me. But the penetrance we'll talk about the next slide. That's what will probably perhaps throw us off. But what would be, assuming 100% penetrance, which we'll define in a moment, assuming that it's like we saw in the previous, what would the chances be of having the disease? 50%. If one parent had it, so presumably the mother doesn't have it, the grandmother doesn't have it, the father had it, it'd be 50% chance. Now, they're using that to illustrate another concept in genetics that's called penetrance. And penetrance is basically, even when, this is where Mendel, his genetics kind of breaks down a little bit. Even dominant alleles, they don't always guarantee the progeny have that phenotype. And the percent chance that they will it's called penetrance. Okay, so in this situation, even people who end up with that bad allele, only 12.5% of them end up having the phenotype. Okay? Question back here, yeah.
What is the question before?
Oh, yeah.
Why isn't it? Because wouldn't it be 3 boxes?
So all that's happening right now, if I were to draw this, it's like the father had the disease, right? Mm-hmm. So a kind of filled in box. Presumably because the mother's still alive and her mother's still alive, I guess that's what they're trying to illustrate, that she's unaffected. Oh, go ahead, yeah.
I think it's because it says both grandmothers are alive and healthy. I think it's saying that the father is likely has one dominant and one non-dominant. Yeah.
Right.
And the mother has 2 non-dominant, so the likelihood that the son has it is only
But they're not
4 alleles.
So the question is, right, so in the likely phenotype here, because this is a dominant allele, is, we'll use B's for here. It's likely the father was this, mother was probably this. Is that the seeming reasonable? Yeah, go ahead.
Yeah. So how you drew the box, it would be big B, little
Mm-hmm.
b, big B, little b. But wouldn't there be 3 boxes with big B's? So
No, I think just 2, and those 2 boxes would be equivalent to 50%. Yeah, I could actually draw the Punnett square there. So just do it like little b, big B, little b. So right here. Does that make sense now? Each of these is 25%.
Okay.
So we're going to add up these top two boxes is 50%. That's one way of solving it. Mm-hmm. Good. All right, so what does this mean for the young man whose father died of it? It means, well, first off, I have a 50% chance of getting it. And then on top of that, on top of just having the genotype for the disease, there's then, only a 12.5% chance that I'll actually express the phenotype of that allele. So it does reduce the chances of actually having that sudden cardiac death. So autosomal recessive inheritance. Now we split the boxes, and individuals that have half a colored in, we call these carriers. Individuals that are solid, that would be an affected individual they're homozygous for the two disease-causing recessive alleles. Okay, so looking at here, we have two carrier individuals. They have 1, 2, 3, 4 children. Again, 2 females, 2 males. And as you can see, 2 of those offspring are carriers, 50% of them, right? 50% of the total offspring. 1 of the offspring is unaffected, and then 1 of the offspring is the affected. And this is the I'm trying to think of the right. This is so ingrained in me. I can't think of the shorthand of this. It's the 1-to-1. That's what they always call it. 1-to-1 ratio. 1, so 25% unaffected, 25% diseased, and then the 2 in the middle is that 50% that is heterozygous, that are carriers. Okay, so be aware of that when you have autosomal recessive. Should be expecting that. Half of your population should be recessive, and then so on. When there's 2 individuals that are both carriers that have children. Now notice, however, it is impossible for a carrier to produce, a carrier or even an affected individual, to have offspring that have the disease unless the partner of that individual also carries an allele. Does that make sense? Mm-hmm. So like over on this one over on the right, this carrier, what ends up happening there, if we did the Punnett square, half of the offspring are heterozygous, half are completely unaffected. Over here, with the affected individual, with the spouse being unaffected, it's 100% carrier. None of them have the disease, but 100% of them carry it because there's 2 bad copies of the gene that are coming, so at least one of those always ends up in the progeny. Okay. Yes, question, yes.
So theoretically,
yes,
if you could map out these genomes on every person who was ever gonna
yes,
mate,
yes,
you could essentially eliminate these if you
yes,
mated the right
yes,
people.
Gattaca.
Theoretically.
Yes, absolutely. So one of the interesting things about Gattaca is that they talk about a modern society, and this is not far from where we're at. I'm not sure if I should bring it up just because you can kind of ethically wrestle with it in your own head. A modern society where it's just very commonplace where you get tested and any potential partner, before you even start dating them, you also want to hear their results. And you want to hear their results not only for their diseases, but I want to know your quotients for your genes that influence your phenotype. Tell me about your intelligence genes. I want to know what your quotient is for your genetic potential of intelligence. Tell me about your any of the other parts of your genes that might affect your personality. What's your percent chance of developing, any sort of personality disorder or something like that? And so it makes a big impact on society. So, good movie, Gattaca. So, just saying. And beware, obviously. Beware of what that could lead towards. Okay, so cystic fibrosis is the most common autosomal recessive genetic disorder. Cystic fibrosis, most common autosomal recessive genetic disorder. And actually, the first recessive disorder whose gene was identified. Francis Collins, NIH, he's now the head of the National Institute of Health and oversees all of the research that comes out of there. He was the one who helped discover the gene here. Recessive disorder. So, which of the following statements is true about an individual who's a carrier for cystic fibrosis gene? Okay, wow. People jumping. So, heterozygous does not suffer from cystic fibrosis. Does that make sense? Heterozygous, you can't suffer from it. Homozygous does not suffer. Nope. Homozygous suffers from cystic fibrosis. If the homozygous is for the disease-causing allele, then yes. But this is specifically talking about carriers here. Okay. Okay, so that then leads us to the third major category, sex-linked traits. So X-linked is the major category here. The gene is going to be sitting on the X chromosome. Of course, we know females have 2 X chromosomes. Males have 1 X chromosome. One important factor about males having 1 X chromosome, they always get their X chromosome from their mothers because they had their Y chromosome from their father, and the sperm didn't have an extra X chromosome in there. There's only 1 copy of each chromosome, right? And so it can only have 1 in there, and so always a Y chromosome. So every single X chromosome that's in a male always inherited from the maternal side. Okay? Females, of course, get 1 X chromosome from their father and 1 X chromosome from their mother. Okay? So if this Brugada syndrome was sex-linked, what would the young man's father's genotype have been? What would that be if it had been X-linked? It's actually not, but let's say it was. So if it's on the X chromosome, he's only got 1 X chromosome, right? So that means that he would have a affected X chromosome, and then of course you have the Y chromosome, and he would basically have a single, whereas the unaffected mother might have had just 2 normal X chromosomes. Does that make sense? Yeah, so the father, if it was sex-linked.
So what was Brugada syndrome? Was it dominant?
Yeah, it's actually autosomal. It's autosomal dominant there,
Okay.
but it is autosomal, right? But now they're saying, okay, let's switch it over. So if we switch it over, it's got to be on the X chromosome. We know the father is XY. He's only got 1 X chromosome. It's got to be affected. So basically, in terms of like writing the allele, you know how before we would say, you know, like here's the dominant, and then if they had, you know, you can't even write this for a male on the X side of it. It would actually be, you'd have a 0. Does that make sense? So like a female could be a carrier, but a male can't even carry. It would either be having the allele and then having nothing, right? Because it's hemizygous, because there's no corresponding, the Y chromosome doesn't correspond to the X chromosome, or it would be like this. The 0 representing there's nothing to compare it to.
And so that's not just for this, that's for all sex.
For all sex-linked, for any sex-linked disorder, when it comes to males, they're either going to have the dominant allele or the recessive allele, but either way, you know, they're going to express the phenotype of that one allele that they've got. So in terms of what the son would inherit, the son would inherit, the good news about this, you guys do the math on this, the son would inherit what from the father? Yeah, the Y. I should have circled that, but yes, should have put a line. There we go. So there's no chance that he could actually pull it from his father. That's right.
So then if he had if the father had a daughter,
guaranteed she would have gotten that X chromosome. Right? Yeah. Okay, so here is here's the X-linked. Notice the little tiny circle in the middle here. This represents a female who is a carrier of the disease. So she has 1 dominant and 1 recessive allele. Most X-linked diseases are recessive. Most are recessive, meaning females tend to generally be carriers. Very rarely are they ever have the disease. They can, if they happen to get really unlucky and they get two bad alleles, right? Two bad alleles, one from the father and one from the mother. But most of the time, they are heterozygous.
So the dot in the middle means the X-linked recessive gene?
Yep, it means that this individual is heterozygous, that it would have both the recessive allele and the dominant. And so then you look at pairing here, what does that result in? Sure enough, as we would anticipate, 50% of the progeny pick up a copy here. When it's the female, picks up one bad copy from mom and one good copy from the father. But here's the male here. The males who are being descended from a female who is a carrier, their chance of getting it is what percent? Yep, it's 50% because they could either get the dominant allele or the recessive allele. They get one or the other. So obviously this offspring got the one that didn't cause the disease, and this one got the one that did cause the disease. Okay, and this one that caused the disease, they only got that one copy of the X chromosome, so they actually have the disorder because there's no extra copy of the X chromosome with a good copy to save them and rescue them. Yes, color blindness is a good classic X-linked disease. That's why far more males are affected than females. Yep, because the opsons are encoded. Some of the opsons are encoded on the X chromosome. Yeah.
How did the left side one go?
This one left side over which one? This one?
The square.
The square. The one on the right here, or on this side down here?
Yeah.
Aha, yeah. So again, we basically just replicated what happened up here. The mother is a carrier, one dominant normal allele and one recessive bad allele.
Oh.
So she's going to give one or the other to every offspring. And so this male must have gotten the normal one, and then this one must have gotten the recessive disease-causing allele. And they only get one X chromosome. And then each of the females that are descended from that mother, again, 50% chance of either one. So this one over here from the mother must have gotten a normal one, and then this one must have gotten the disease-causing one, but it's not causing disease because her father also gave her an X chromosome, so that one's there. Does that make sense? And we're assuming the father has a normal X chromosome, so there'd be a normal X one across the board here.
So the sons are the ones who are affected?
So the sons are much more commonly affected, that's right. They're not exclusively. If we set this up, we could actually produce, there could be a female that would have two. Set this up, yeah, I guess, yeah, you wouldn't want to arrange marriages and all this, what?
No, we're laughing because I think there's a typo in the picture. The far right should be in the middle row, the middle image, unless we have two males marrying.
Oh, hey, that's it, yeah, very interesting. And producing children who are genetically related.
Welcome to Union College.
Okay, hey, whoa. So there you go. That is interesting, yes, that is a significant. We did not go down the right side of that. That is true. So let's just make that a circle. Be a little more realistic there. A little more realistic. That actually is worthwhile going down that side. If we change that, let's say we have an affected male, so we know that they are affected, so they have the recessive X, and of course they have a Y chromosome. They pass the Y on to any male offspring. So there's the good news is 0 chance of these males being affected, right? Because they're going to guarantee get the Y from the father. But on the females, there is basically a guaranteed chance, right? Every single female is going to be a carrier at least, because the father's going to pass on that exact X chromosome onto each one, the recessive allele. Yeah. Oh, so let's make her a carrier. So let's change the gender and now change the genotype here. So let's put it on like that. Is that what you're saying? Kind of like that? Yes. Okay. So now we're getting serious now. So the females now, finally you might actually see a female who would have an X-linked disease, right? Because they're guaranteed getting one bad copy from their father, and then if the mother's a carrier, 50% chance they get the bad allele from the mother, so one of these could actually be affected. And then, of course, the other one would be a carrier still. I mean, yeah. Good questions. Whew. Yes. Yes.
On the two males on the bottom left, you put two X's. So
Oh, that's a problem. Yes, yes. Oh, I'm sorry. I apologize. That is confusing. Yes, I should have put Y's there. Yes, this has been the normal big X, and then this would have been the recessive that they got. Yep, thank you. That's also true. Yes. Someone like me apparently made the
On the right, so in that situation, if you had a carrier female with an affected male, both of the sons would be guaranteed to get it as well, correct?
Oh, yeah, the males. Yes, these males. Well, let's see here. The males would still get the Y chromosome, so that's okay, right? And they would have a 50% chance on this particular, right, they have a 50% chance here. So this one would potentially get it.
So you have 50% affected with 25% carrier, 25% unaffected.
Right, yeah. It's pretty far down the rabbit hole, doesn't it? Yes. Yes. Okay. So let's do a simple, straightforward question here. If an unaffected mother, I'm going to write this. So unaffected mother carries one normal and one mutant. Okay, so I'm going to go ahead and write it in there. So one mutant and one there. Okay. On her X chromosome, what are the chances, chromosomes, because you got two, right? So there'd be one normal allele on one X chromosome and one mutant allele on the other X chromosome. What are the chances that she will transmit the defective genes to her son? So it doesn't really matter what the male is here. We're just asking, okay, what are the chances for any sons that come of this? So what do you think? Yeah, 50%, because she has two copies. One's bad, one's good. Each male is only going to get one of those, and sure enough, it could be one or the other, and so it's going to be 50%. Okay. All right, so if you have an allele for a trait, do you display the trait? So obviously, here's the answer. If it's recessive and you only have one copy of it, then no, you wouldn't display the trait, right? It kind of depends on the situation. If it's a dominant allele, yes. If you have at least one copy of it, you're going to display that trait. And then the sometimes answer is, remember that idea of penetrance with that autosomal dominant disorder, where even if you have it, there are some diseases where it doesn't even guarantee that if you have the genetics that it'll actually present the actual phenotype syndrome. Okay. Now, I don't think we have much on genetic imprinting that I need to cover. I'm just looking over your learning objectives, make sure I hit those. So I think I may skip over that. We've done X-linked. I think we need to go on, make sure we do Down syndrome and teratogens and environmental influences. Very briefly, mitochondrial DNA. I'm sure you're aware, maybe you're not aware, but mitochondrial DNA is always inherited from the maternal side, and that's because sperm, as they join with the egg, they do not donate any mitochondria, and mitochondria are separate from the nucleus, so their own little organelle, and that own little organelle actually has its own little DNA genome inside of it. Each mitochondria's got its own little DNA sequence inside of it, and mitochondria separately replicate themselves. They duplicate, right? They grow. Well, the egg is going to have dozens of mitochondria in it, and the resulting zygote, whether it's male or female, all those mitochondria come from the maternal side because the sperm didn't bring any. And so actually the sperm can bring just a few, but they usually get washed out in terms of, it'd be like 1 versus 30-something, and over replication, very few of those will end up, making a big difference on the phenotype. So mitochondrial diseases, generally speaking, they're coming down through maternal lineages. Okay. Now, this slide here is not for you to memorize all of these particular chromosomal problems, but more just to illustrate that chromosomes can get all kinds of twisted up and have a variety of different structural abnormalities. These are big abnormalities in terms of we got large pieces of a chromosome that is kind of messed up. Well, I don't want to go down into too much detail here for you, but basically, if you just look at each of these items, we'll just do deletion perhaps, parts of the chromosome are either being lost completely or swapped onto the wrong chromosome. And that can end up causing pretty substantial damage because you're either missing genetic information or that genetic information might be on the wrong chromosome, and it might be controlled inappropriately then because it's on the wrong chromosome, and the control mechanisms of that chromosome are perhaps controlling it, and it ends up causing a problem. Yeah, I would not worry about this particular one here. I want to go on. We got to do, yes, our aneuploidies, including the trisomies. This is the key thing you can't get out of here and not know. Aneuploidy just says there is an unequal sorting of chromosomes during meiosis. So normally we sort it out, and for every 4 eggs or 4 sperm, each of them gets 1 copy of each chromosome, right? Well, sometimes not. Sometimes they'll actually be missing a particular chromosome. Okay? So Turner syndrome, this is usually XO, is what it's called. This is where they don't get a Y chromosome or they don't get an X chromosome. They just have 1 X chromosome. Does that make sense? And so that's because in the sorting out, the X or the Y chromosome got lost, and the chromosomal machinery to move chromosomes messed up. Very rare and not much compatible with life is where you can actually have every single chromosome have an extra copy. So it's not in humans really. You don't see something running around with trisomy. So this would be a trisomy of every single chromosome. Polysemy is something you can see in some sort of in certain plants and other things, but in humans you're not going to see much of that. It can happen, but it usually just arrests development in utero. It doesn't result in normal life. Now, trisomies, there are 3 trisomies that are, and the most common trisomy by far is Down syndrome. So trisomy 21. The other 2 are Patau syndromes 18 and Edward is 15. These are substantially less common. And then the rest of the chromosomes can have trisomies, but again, they're usually lethal. They don't result in an actual person being born. But trisomy 21 Down syndrome, absolutely, very common, relatively common, and still produces a very viable embryo. So again, 21, and if you look in here, instead of having 2 copies of that chromosome, you got 3 copies, and that means either the egg or the sperm donated an extra copy. Okay? This results in a number of challenges. When you have those extra copies of the genes, it will change some of the things. Some of the things it changes are just kind of the way someone looks or phenotype. It also changes a number of things for medical conditions. For example, most individuals with trisomy 21 Down syndrome have heart challenges, intestinal malformations. They are much more likely to have leukemia. They're actually much more likely to have, and they don't list this here, but much more likely to have Alzheimer's disease because they get 3 copies of a gene that is linked to Alzheimer's, and so they get an extra copy to increase their risks of developing Alzheimer's as time goes on. Yeah?
Is the congenital heart disease usually what causes them to
Die?
life expectancy?
That's a good question because there is shorter life expectancy. Off the top of my head, I don't know. They have a number of health problems, and I imagine some of that stuff is additive, and it could be that the overall probability, but you're absolutely right, there is some reduced lifespan. Yeah. Okay, let me make sure. Turner syndrome, this is what we saw earlier, the XO, again, of the individual who's missing a Y chromosome, and then Klinefelter, this would be XXY, where they get an extra, they have both an X and a Y and an extra X. and it results in some other challenges there. Last point to make as you run out the door, there's lots of diseases. In fact, most diseases fall into this category where there's multifactorial, where it's both genetic and environment. Classic example is cleft palate, which there's some genetic influence. There's also environmental influence that we're aware of as well. And I'm going to leave you with